most exciting projects. I mean, there's a plethora of projects we're working on here at the moment on the cancer team. Certainly, we just published a pancreatic duct adenocarcinoma paper, which showed for the first time that we can use these microphysiological systems, our organ on chip, to really show also immune evasion into these tumors, see that we can treat these PDAC tumors with drugs and see how they are going over a vascular barrier into the tumor. So this is very exciting, but also some other work where I'm allowed to look at neoangiogenesis and kind of build vascular networks in a chip. And this is all very exciting work and amazing what one can recreate in such a small organ on chip.
Definitely online readouts, online monitoring of organ on chip models to see in real time what happens inside the chip with the model, and to see really in short time intervals effects and not just rely on endpoint measurements.
The TANA project, although it has been a bit challenging so far, but it's really exciting because it's now offering a new perspective, new avenues for the treatment of long aspergillosis. And it's a disease that we know it can be very fatal for immunosuppressed individuals.
The most exciting project I'm currently working on is based on the intestine on-chip model. So this is what I basically do during my dissertation thesis. But we are also currently developing this intestine on-chip a bit more by including primary intestinal epithelial cells, and then in the next step design a completely primary-based or stem cell-based intestine on-chip model. And this model is not only primary cell-based, but will also include a lot of biochemical but also electrical sensors to really have a real-time measurement inside of this organ on-chip model. And furthermore, also to include oxygen gradients for host microbiota integration.
It's the project that I'm currently working on. Actually, it's the chip that allows us to cultivate 3D vessels, real network of 3D vessels. And I think with this opportunity, we can create a really complex models for human physiology. But also for me, it's really interesting how from the suspension of cells we can create such a complex structures as 3D vessels, and then, for example, combine it with spheroids. So the spheroids are actually vascularized.
The most exciting project I'm currently working on is the further development of our liver-on-chip model, not to have it only for the human, but also for other species, like, for example, a rat liver-on-chip model. This would also enable us then to compare the data which we received from animal models, to compare this with the organ-on-chip technology from other species, and also then comparing this data better to the human physiology at the end.